to be doing is taking a look at some data from the Red Rock Reservoir. It's a little, it's actually not that little. It's a decent sized reservoir for Iowa that's about, I don't know, an hour southeast of Ames. And the Iowa DNR has collected data as part of their ambient lake monitoring program on this reservoir for quite a number of years. And you're going to go to this website, and this is a really good practice of taking a look at real data, because real data is noisy and messy and difficult at times, and use that data to answer some questions and understand some outcomes that are going on in this reservoir and what's happening. So the first bit is basically going to this website and getting the data and kind of just looking at the data, organizing it as you go. You can use, I think, my suggestion for you, Tyler, is to just stick with Excel. Alvaro, have you worked in MATLAB or R much as in more of your advanced courses?
I hate MATLAB because I don't understand it, but I'm trying to learn R.
So this is a data set that is not that intensive. It's not that large. So working in Excel works fine. I try and give my seniors and grad students, so being that you're a little further along and actually in both of those categories, this would be a good data set to practice using some R code if you would like to.
Okay.
So I might suggest you try it, but it's totally up to you. There's no like preference or whatever. We're going to do some looking at nutrient limitation in this lab, and there's a reference to a paper here that I will post in Canvas that you can take a look at. But the idea really is that you're going to look at nutrient concentrations, and the two that we're going to look at are nitrogen and phosphorus. And remember, we talked about the many forms of nitrogen, so you're going to have two forms of nitrogen in this data set, both ammonia and nitrate. I'm sorry, this is probably a little bit tiny. Let me make it bigger. Is that better? Good. So when you look at that data, you'll see these two forms kind of listed, and I can show you what that spreadsheet looks like in a little bit. But essentially, the way we report nutrient concentrations is normalized to the main element of concern. In this case, it's nitrogen. So rather than when we report this as mass per volume, most of the time you would use the molecular weight of the compound, the molecule, whatever. But when we report this in a water quality context, we report it as milligrams N per liter. So it's normalized to the mass of nitrogen, not normalized to the mass of ammonia or normalized to the mass of nitrate. Why that is important is because then, since they're in the same unit space, they can actually be added together directly. You don't have to do any weird conversions. So if you remember back from chemistry, you can't add concentrations together of different elements unless they're in a molar ratio. That was a classic way we would do that. We normalize them to molar concentrations, so that way you can add them together if you needed to kind of look at their cumulative effects. When we do this, we've normalized them to the same molar mass, so the mass of nitrogen. That allows you to do that. So that's what that all means. We can add these directly because they are expressed as milligrams per liter N. So what you're going to do is you're going to get all this data, and you're going to have nitrogen data as ammonium and nitrate, and you're going to have phosphorus data as orthophosphate. We'll talk a little bit about phosphorus today, and the form that we measure typically in water bodies like lakes and reservoirs is a filtered sample. So we collect some water and we run it through a filter to get rid of any of the algal biomass or other silty clay kind of materials, any of those particles. What comes out the bottom side, that filtrate, is what we measure for SRP, soluble reactive phosphorus. We, as a discipline or as a just convention over time, it is really impossible to say that is all PO43 minus, because some of the larger molecules of organic phosphorus, so you think like we talked about carbon and how it has all these compounds of nitrogen associated with it. Well, a lot of phosphorus exists similar ways, right? ATP, adenotriphosphate, right? There's lots of phosphorus molecules in cellular, components. So a lot of those molecules can be small, but they still pass through that filter. And so nitrogen doesn't really behave that way. It either exists as a larger organic molecule or it kicks them off as these ammonia and nitrate species. Phosphorus is kind of weird, so we basically operationally define it as anything that passes through a 0.45 micron filter. The chemistry is the same, though. We're taking all of that phosphorus, whether it is existing as phosphate or as a very small organic molecule, and converting it through a digestion process and basically measuring that colorimetrically. And we measure the color change in the water based on some chemistry, and we can relate that to the phosphate concentration. So to kind of recap, you're going to have data on this reservoir of the nutrients, nitrogen and phosphorus. And you're also going to have data, to come back to part A, about the ecological response of the of the reservoir's biology to those nutrient concentrations. So like we did with metabolism, you're going to have dissolved oxygen over time. So just like we did with our metabolism lab, you will have that data. So those are going to look kind of like, you won't see this diurnal cycle because the frequency of measurements at Red Rock Reservoir, you will note, is like 3 to 4 times a year or once a year or something like that. It depends on the frequency at which these data were collected. So it's not a diurnal signal. It's not even a daily signal. It is a snapshot, one at a time. We have it for a long period of time. And at the same time of measuring dissolved oxygen over this period of record, they've also measured chlorophyll A. So chlorophyll A is essentially an indicator of algal biomass. So we do these samples the same way. We collect a water sample and we filter it through a little paper filter. For chlorophyll, though, we actually do it the other way. We actually analyze the captured green algal biomass on that filter, and we analyze that I can tell you how we do that, but it's a pretty standard method. So then we know how much water we filtered, we know how much biomass of chlorophyll or plant material normalized to this metric we can measure of chlorophyll, and then get kind of a surrogate for how much algae are in the water. So we've got chlorophyll as a growth response, a biomass indicator. We've got dissolved oxygen as an indicator of that metabolism signature, right? And we've got nutrients, nitrogen and phosphorus. So similar to what we did last week, where we just had kind of the oxygen signal and we were kind of relating that as an inference to production and respiration. Now we're going to kind of take it at a larger scale and see how if we measure biomass directly and we look at the oxygen signal and the nutrients, can we get an understanding of what is happening in that water body that is changing water quality, and what is the primary cause of changes in algal production. The biggest indicators of lake health are typically 2 things. One, excess algal production. We don't like a lot of algae because it's not pleasant to swim in. It's not good for other organisms that are in the lake if there's a lot of algae competing for causing dissolved oxygen to plummet, like we talked about with the Gulf of Mexico hypoxia last time. So that's one indicator of poor lake water quality. Another indicator, which we are not going to explore in this particular lab, is bacterial load. So if you think of a beach closure, the beaches are closed because they have high levels of a pathogen or bacterial load. So that's usually measured as E. coli. So there's either an ecological problem that we think of with the eutrophication and all of that, and that does kind of cascade to other human health concerns as well, or maybe a direct human health risk with high levels of bacteria. So the bacteria stuff we're going to put on the pause button for a minute, but the other big reason is this. And so we're going to really explore what kind of data we collect and how we can use that data to understand what's happening. Questions so far? Does that make sense? was a lot of information. I saw Tyler frantically writing, and I saw Alvaro very gently nodding and listening. So, do you have any? Does that make sense? Is that clear?
I might have to go back and watch the recording for a few things, but
Okay,
yeah, his
that's
the concept I'm
makes
understanding now.
sense. Okay, great. Alvaro, good.
I think questions will arise once I'm working on it.
Okay, so I'd like to pause, and while you guys, we'll go back into the presentation, and we'll go kind of continue on with the nutrient data, biogeochemical cycle stuff now, and then I'll put this up on Canvas. It'll take me just a second, and let you get to that data set, and we can work on that. So I'm going to stop kind of teaching at about 30 minutes from now. So at about 10:20 or so, whenever we have a good stopping point, 10:15, 10:20, we'll pivot to let you guys work on this for about 30 minutes. But I'd like for you to work on it while I'm here so that I can help get you over some questions that might emerge, because this is probably a little bit new for you, Tyler, especially because this is a data analysis, data science kind of project or assignment, and I want to make sure that you have all the tools you need to get going.
Sounds
Sound
good. Thank you.
Perfect. Okie doke. So where we left off last time was the nitrogen cycle. And so I am going to, oops, need to share my screen before I do that. You can go away. You guys see that? Okay, awesome. All right, so what we did last time was kind of drew this on the board. So I just want to kind of go back over it and make sure that we have all the bits and bobs together and we're clear about where things are coming from. Nitrogen is complex and has lots of forms. It exists in atmospheric forms, gaseous forms. In the atmosphere and gaseous forms, it exists in dissolved forms of nitrate ions, ammonium ions. It exists in solid states as organic nitrogen, primarily from waste decomposition, biomass decomposition, humans putting fertilizers onto soils. Sometimes those are as an organic fraction, like we talked a little bit about with like urea. We use at one of the farms I work at, we apply fertilizer as UAN. You might hear that term used quite a bit. Avara, what is that? Do you remember or no? Have you heard it before? Might be an American thing. That acronym stands for urea ammonium. So U is for urea, AN is for the ammonium. And so it's a mixture of ammonia, which is readily available to the plants, and urea, which is a very small organic compound that is gonna stick around a little longer and not get rapidly converted to nitrate through this nitrification process. So remember, we talked about how microbes work along this continuum. So this little zoomy arrow at the bottom, this stuff happens in soils. It happens in wetland bottom sediments. It happens in all kinds of riparian areas, but it doesn't really happen in the atmosphere. They need microbes to do this process. It doesn't really happen in water that much unless there's a lot of particles that these organisms can attach to. Because it's microbial driven, it is predominantly happening in soils, and those soils can be wet or more dry. The trick is that understanding where they happen in that soil profile. So if I were to kind of separate this into two pieces, this doesn't do as good a job. I'm gonna have another slide that has more of this. Let's see. Oh, look at that. I have another one that is helpful. This is more complicated, but this is a slide that my student put together as part of her dissertation work on nitrogen cycling in wetlands. And you'll see lots of arrows and a few more complicated pieces here. So you'll see ON now, which is organic nitrogen, and you'll see a few other gas forms, a few other ionic forms, and we'll talk about these a little bit because I think they are kind of helpful to the conversation and understanding where these processes take place. So because ammonium to nitrate, if you look just at this little arrow to this little bubble to this little bubble, you're adding an oxygen, right? So that is a cue that you must be in an oxygenated environment in order for that oxygen to be available to get added to nitrate, right? So nitrification only happens in oxic conditions. So it doesn't really happen that much in wetlands because the only way oxygen gets into these surface soils is through leaching from roots, because roots have to respire, remember? And if roots are doing that respiration, they're bringing in water, they're bringing in nutrients, and those very sponge-like roots also leak out oxygen. So you can have nitrification happening in the root zones of these wetland plants because of that leak of oxygen. Where we see it most in agricultural lands is in surface soils in fields. So I'm going back to my other nitrogen cycling picture. In this soil fertilization space, we see that groundwater table is deep, right? We don't have any, might have some water in those pore spaces, but it's not saturated. There's lots of space for oxygen to exist in the pore space, right? So there's oxygen there, and there's a lot of fertilizer applied, or even a manure or other organic forms, so that ammonium is readily available, and so is oxygen. So in that case, nitrification proceeds very, very rapidly. Nitrification is an energetic process, meaning energy yielding process. So these organisms will continue to do that. They'll get energy from it because they've got all these substrates around. Okay, questions about that? So nitrification, energy yielding, needs oxygen. Those are the two key things to remember, okay? There's all kinds of stuff happening over here. I'm gonna come over to that later, but looking on this side over here, this is just a blow up of the thing we talked about last time. Denitrification goes from nitrate to N2. A lot of the nitrate comes from overlying water. It doesn't necessarily come from this process that much, particularly in agricultural lands. Much of that change of surface fertilizer to ag soils creates this big pool of nitrate in our landscape, and that's in the land, the terrestrial land. It rains, and that nitrate comes into water bodies like wetlands, like rivers, like even little puddles in fields, right? So that's what we're saying here is this inflow, this big arrow she's showing here of inflow of nitrate by diffusion, kind of just moving with a concentration gradient into those soils. Microbes will take this nitrate and use it as the fuel for their metabolism. So most respiration uses what as its electron acceptor?
What is SON?
I'll come back to that. Answer my question first. What is our electron acceptor for respiration? What do we need for a microbe to decompose some organic matter? I'll give you two choices. Is it carbon or is it oxygen?
Carbon. No.
That's the electron donor, okay? Electron acceptor is oxygen.
Okay.
Okay? So our electron acceptor is oxygen that is taking those electrons, shunting them around and making energy. Microbes can switch and use this heavy oxygen. There's a lot of oxygen there is a good way to think about it. But nitrate is also a very energetically favorable electron acceptor. When we talk about other metabolism in a little bit, there's lots of ways in which microbes can do this, a handful, not millions. And they decompose organic matter all doing this process, but they can kind of get more or less energy depending upon how efficient a reaction is, right? And so the electron transfer for nitrate is not that much worse than it is for oxygen. So nitrate as an electron acceptor, particularly in landscapes like ours where nitrogen as nitrate is super plentiful, will proceed very rapidly. So this process will happen a lot. We have a lot of nitrate. The reason that my student kind of highlighted this, and I'm just going to give this to you as like a little aside, you don't need to really dig into this too deeply, is that each of these little arrows, and this was part of her research, was trying to understand all these connections. Nitrate doesn't go straight from NO3 minus directly to N2. It goes through many steps. And all of these steps can be done by one type of microbe, but not typically. So they're done by a community of organisms that live in this sediment ecosystem. And all of these reactions are facilitated by enzymes. So different enzymes are coded to make these reactions happen. So remember enzymes, they're like a little gatekeeper, and then you get your little key plugged in from whatever production happens. And so then that enzyme becomes activated, and then that enzyme can facilitate or catalyze a chemical reaction. So all of those enzymes cost a little bit of currency for these, and in the form of energy for these microbes. And so as it proceeds, you can start to get a kind of a cascade effect, right? You go from nitrate to N2. What is kind of cool and a little bit unfortunate is that as you move along this pathway, these first three are all dissolved ions. They will stay dissolved in water. Once you get to N2O, however, it tends to be, it is a gas that is released rapidly. This is actually a greenhouse gas. And as that process happens, it just costs the microbes more and more to make this last step happen, nitrous oxide to N2. So if there's a lot of nitrate around, they don't always do it. So when there's nitrate-rich ecosystems and nitrate-rich environments, you can tend to get more nitrous oxide production because there's just more nitrate around, and the microbes are like, why would I spend all that time, energy, etc., making more enzymes that I don't need to, because there's an ample supply of nitrate on the backside. So this was a lot of her research, and she showed a lot of interesting connections between how this proceeds, the efficiency of this process, and how it changes in different planted wetland ecosystems. So plants kind of matter for a lot of reasons. Carbon that's leaked out of their roots, oxygen that's leaked out of their roots, decomposition, etc. So back to your question, Alvaro, I didn't forget about you. What is SON? So it's in her version of this, it means soil organic nitrogen or soluble organic nitrogen. The idea really is that this organic, I think it's soluble. It is. Sorry, took me a minute. SON is often soil, but I'm pretty sure in this case it's soluble because she's talking about particulates up here at this part where it's like plant residue as organic ends. This is particles, and then it can kind of leach out and decompose and still be organic. It's not into the ions yet, but it's now kind of more dissolved in the water. That can be much more stable, but it can be turned into ammonia, and it can be turned into even smaller organic nitrogen compounds that should still probably say SON and then be mineralized that direction. So there's a lot of stuff happening on this graph, but I think it's helpful to show kind of how these things happen because this oxygen is going to drive a lot of these processes. Oxygen from the roots will drive nitrification. Oxygen from the atmosphere and surface soils that have a lot of pore space will also drive nitrification. You need to have that oxygen be used up by the microbes for this process to occur. The thing that's kind of confusing or maybe a little bit misleading on a graph like this is that it looks like these processes are happening way far away from each other, and that this is happening in this place, and then over a meter over this way, this other process is happening. These gradients persist on molecular scales almost, right? They can be changing on the micro meter scale or even the millimeter scale, right? So you can have groups of these organisms doing nitrification, and right snack sandwich right below them where the oxygen is gone are the denitrifiers that are doing this other process. So this is a little bit misleading because of the spatial separation, but these things happen quite close together. That was a lot. How do we feel?
Good.
Clear as mud?
I was curious what PN going into the soil through deposition it represents?
So this PN, P stands for particulate. So in her, we sometimes call it particulate. Sometimes we call it organic nitrogen as particles. That's particulate nitrogen. Often you'll see it in data sets like the Lake Erie, or I'm sorry, I'm not doing Lake Erie anymore. The Red Rock data set, you might see it listed as total N. So that is the stuff that includes all of this, the nitrate and ammonia, and anything that's particles. So it's like called a total N. So if I had a little, can like try to use this little, let's make it pink and I'll make a little brighter. Can I do this? So this guy equals total N. Look at that. Writing with a mouse is hard. You need a pen. So that like grouping is what you'll see in your data set. Good question. Anything else? Okay, we're gonna keep going because I want to introduce, oh darn it, now I have to erase that thing. How about just my eraser? Okay, cool. So just to quickly summarize the nitrogen cycle, we've got N fixation, N2 turning into NH4. Then we can have that being assimilated by some sort of organism, but whether it's a plant or a microbe. That goes to nitrification by, again, facilitated by a group of bacteria that take this ammonia and turn it into nitrate. Here is what that reaction looks like. So this is chemoautotrophy. So we're generating energy, but it's not using decomposition as a way to do that. It's going to have to get carbon into its system as in another way, but basically we are taking energy out of this reaction and using it as the chemistry is our energy source, not the sun. So we've got ammonia going to NO2, and typically we call these done by this group of organisms called nitrosomonas. It's been isolated years and years and years ago. Now we know that there's a whole group of different organisms that do this, but it's still kind of useful to kind of use these two groups of organisms because it does illustrate the fact that this isn't done by one single organism. Then this other group, Nitrobacter, takes it from NO2 minus all the way to nitrate. But each time we're adding oxygens. And so here's the final chemical reaction. I'm not going to worry about DNRA. This is a study, a process that we work on in my lab a little bit, trying to really unpack all the components of the nitrogen cycle. That stands for dissimilatory nitrate reduction to ammonia. It's another process facilitated by microbes. One of the things that you have seen already is this figure do you remember this picture? I love it. I use it in all my slides. You'll see it eventually, again, Tyler, in my next class. But the reason we care about this conversion of nitrification is that nitrate is represented by these little orange guys. Ammonia is these little blue purple guys. Most humic substances, so that organic kind of detrital material, we call that humic matter, or clays, small little clay particles, have, for different reasons, their net surface charges are negative. So that means they can really strongly attach ammonia to their surface. It's not a reaction chemistry, anything like that. It's actually just absorptive ion exchange process, right? So it's just kind of holding on to that loosely. But it holds on to it, which is great because then plants have it available. But like we just talked about, ammonia doesn't stick around because in all these pore spaces alongside this orange kind of negative charge species as nitrate also lives oxygen. So it's taking all of these little pluses and turning them into minuses. Those are the plus ammonias, turning them into the negatives for nitrate. Now that negative and this negative are like two repelling ends of a magnet. They don't want to stick anymore. So what happens is that nitrate lives in the pore space. So we've got nitrification happening because it's energetically favorable and there's a lot of ammonia available. When we generate nitrate, and nitrate, what does it need to denitrify to turn back into N2 gas and be removed from our environment? It needs an oxygenated as an anoxic environment, sorry, the other way. So if we don't have that anoxic condition, it's going to just sit there and it's not going to change. And so it's just going to like accumulate in the pore space, right? So what happens when it rains is what this blue line represents. A rainfall infiltrates into the soil profile, and all of that nitrate comes along for the rain. So that gets flushed out of our system. This flushing can happen as nitrate flushes out of soil pore spaces. It can go into tile drainage, or it can actually get kind of re-emerge at hill slope seeps or different kinds of slope changes and come back out as surface runoff. In order for this process to happen, denitrification, that's the one that we need those oxygen limited conditions. So it doesn't have to be completely gone, but it needs to be mostly gone for denitrification to proceed. Okay, we're going to talk a little bit about this in greater detail, but the summary is that we need, this is also, like I said, a metabolic process, so we need denitrifying bacteria. We also, fungi do this as well. We need those organisms present in our system. Newsflash, they're everywhere, so that usually is not a limiting factor. We need a lot of nitrate. We also have a lot of that in our agricultural landscape broadly. We need organic matter, and we need good quality organic matter for it to proceed rapidly. So we need a total amount, so that's what that F means, function of, so think back to like your math days. The organic matter needs to be both enough quantity, so the overall amount, and its composition or its quality. How complex is it? If it's a very simple carbon, it's much easier to get energy out of. Same as regardless of whatever metabolic process is happening, whether it's denitrification or aerobic respiration. But we need that oxygen to go away. Otherwise, those microbes that are doing this process will switch. So denitrifiers are a little clever group of organisms. They have evolved to have machinery, those enzyme production mechanisms in their DNA to use oxygen when it's around. And then when oxygen is low or gone, they will switch and fire up the enzyme making machinery to make nitrate reducing enzymes. So they're the same group of organisms. So you can imagine as oxygen changes, they're going to change their metabolism. So oxygen has to go away. But the nice thing is that these organisms are what we call facultative. So that means they can switch. And so they will live in these sediments and they will switch over to nitrate and do that process. So this little cartoon from Iowa Learning Farms, I think, kind of summarizes that nicely. We have external nitrogen loading. This is usually from fertilizer application, right? Ammonium turning that turning into nitrate in this landscape. Geez, Pete. And releasing N2 gas. So nitrate is our terminal electron acceptor. Dang it, this is our equation now. If we kind of put this all a bunch of, I showed you this in various simple forms where we have some sort of organic matter, organic compound, plus a nitrate turning into N2 and CO2. And we got some waters that have to come in here because it's got some hydrogen ions. Okay. Any questions about that? You good so far? Okay. Tyler, you good? Okay. So I'm gonna just skip over this. Just another similar summary figure. So what I want to not forget about is trying to link this to multiple cycles and why I keep talking about this as like redox and what does this all mean. So I'm gonna hit escape here just for a second because I'm curious how far I can go for I want to make sure I get to the phosphorus cycle before we're done. Yep, we should have time. Okay, cool. So we'll spend just a few minutes. This is an idea that is a little more advanced, so I'm introducing it, but I'm not going to go into great detail about it. I alluded to this with these two processes. Reduction of oxygen, what is that? What have we been calling this reaction?
Oxidation.
Yep, oxidation. We have not used that term, but what is it? What have we been using? You know, this is only the half reaction of the reduction part. The oxidation part is on the other side. What is the oxidation part? What are we oxidizing? carbon to CO2, right? So go back to this little figure or equation. We're oxidizing this carbon into CO2. And we're reducing nitrate into N2, or reducing oxygen to water. Okay, so these are the half reactions. And this is like a little more advanced chemistry and how it's looked at, but it is a helpful tool, I think. So this is respiration. This is simply aerobic respiration. Okay, this reduction of nitrate, this is denitrification. And then as we go on down the line, we can use other electron acceptors. So we go from oxygen to nitrate, manganese, iron. We talked a little bit about iron and rust formation. That process is another redox process. We talked about that before. You guys were like, yeah, I got some redox. I remember that a little bit from chemistry. That is another one. That is, we don't talk as much about manganese because it's not as prevalent in the environment as iron is. And so those are, manganese reduction is sometimes not as important. But what I want to draw it looks like my lines got a little messed up over here, but this last last number here on this last column is called the Gibbs free energy. And what that is a number that tells you the amount of energy that reaction yields based on its electron transfer. You might think, well, this has 4 and this has 6, so why is that difference? It's related to the number of nitrate ions, right? So there is a little bit more electrons transferred, but we have 2 nitrates in this process to make 2 N2s. But what's very cool is by measuring Gibbs free energy, and people have done this long ago and have done it with very sophisticated isolating methods, is you note that this is about a minus 30. This is almost about a minus 30. So that's why when I say they're pretty energetically compatible, oxygen is better, but not by much. So nitrate often can exist in some of these environments. But then as we go down, we get pretty low pretty quickly. Sulfate reduction is prevalent in marine environments where there's a lot more sulfate around and much less nitrate because we've lost it along the way to the ocean. Talk about that later, but anyway, sulfate reduction can be a lot more common in coastal environments. And this last one, how many of you recognize this as a process? So what are we making? So this is methane generation. So we call this methanogenesis. So this is kind of one of those very low energy yielding, slow processes, but can persist quite unabated in low oxygen environments. So we'll talk a little bit about that. So this is just a summary. I'll let you read this on your own, but we basically go from aerobic oxidation to denitrification to sulfate reduction and methanogenesis. We go from high energy yielding all the way down to low energy yielding. This methanogenesis is really common in wetlands. So we get a lot of methane production in wetlands, not so much in soils and fields unless they're flooded for a long period of time because it doesn't yield very much energy. So it's a relatively slow process. It has to be organic rich, which many of our soils are, not all though. But it has to have it has to have no oxygen for quite some time. And typically in soils, this denitrification dominates. So I'm going to show you this little graph. I usually have students go through this, but so what I'm trying to show here is if you're thinking about a flooded soil, what's going to happen? And so we're going to trace different elements along a gradient of flooding. So this is where we're going to kind of stop with this stuff. But I think this little, hopefully you can take a look at this after time and you can kind of follow along with this. So this is a flooded soil, starts out oxygen rich, and we keep it flooded for a week and we see what happens. We measure what's going on in the water column. So generally what you're seeing on most of these is this concentration, this concentration is here on the x-axis and the time is over here. So let's just trace some of these. And hopefully this kind of tracks with what we've been talking about. So oxygen starts out high because it's oxygenated to start with. And then it quickly gets depleted because there's not a lot of, this is a still pond. It's like a little depressional pocket in the soil, right? Think of our flooded fields in the spring. We got our big field happening, and you see these little persistent, puddles that just kind of don't drain quickly. So that's the kind of stuff we're looking at. So this is one of those depressional spots in our field. Yeah, oxygen's going to go away after about a day. There's none left. All those microbes are using that up for their metabolism. We have a lot of nitrogen around. It does the same thing, but it doesn't start to go away until the oxygen is gone. So nitrate stays elevated from its initial starting point to here, and then it goes away, and it goes to zero around a couple days. And this timing can vary, but it's a good illustration. The next electron acceptor in our ladder is manganese. It looks high here, but it's actually not that available. But you'll note that this easily reducible manganese goes down, and this exchangeable manganese that it's turning into goes up. So if we go back to that equation, you would see the reducible manganese turning into exchangeable manganese. Let me go back to that real quick. And it's not wanting to participate. So we're going from this that's usually attached to an oxygen going into this exchangeable version that's as a 2+. So Mn3, into Mn2, similar thing here, Fe3 into Fe2. So we've got that changing here. But I think what's more also interesting is that once that goes away, you start to see iron 2 being produced. So that is that iron reduction happening. So now you've got, instead of oxygenated iron, that rust particle iron that's associated with iron hydroxides, that is getting dissolved and it is being used for energy, and now we're producing dissolved iron as iron 2. Another indicator of kind of what we call redox potential is this EH. That's a redox potential number, and that's just kind of going down over time. Okay, make sense? The things to take away is that these happen in sequence, and they happen in pretty predictable ways, right? We can really, this is real data. It's old, from Turner and Patrick, 1968. This isn't new. Nothing new under the sun, right? So this is something that we've known about for a long time. This is data that's been collected for years, and it's really helpful to understand these processes in their progression and what causes, rates of these things to change. So yeah, this is all, we know a lot about this stuff, but there's still a lot that we are understanding as we go along and learn more about different places and environments. I'm going to skip this, but you can come back to this idea, but methane production in wetlands can be a pretty rapid process, and you can actually have kind of both methane being generated under low oxygen, and you can have methane being oxidized and converted back to CO2 in the root zones because of this oxygen prevalence. So you can get this kind of stuff happening. So I'd like to talk about phosphorus next, and then we'll go to our homework. It's 10:20, but I want to give us just a couple minutes, so we'll get to this in just a second. So we talk about phosphorus. I kind of introduced that at the beginning of class related to this assignment. Sorry, that picture is terrible. But phosphorus is often what we see as a limiting nutrient of growth in freshwater lakes. Remember, we looked at those plots in metabolism, GPP versus ER. We looked at some graphs of N and P and how they're connected, and they're that graph of N versus P is really predictable because biology have evolved to this kind of typical ratio in a lot of our environments. What happens with phosphorus is it's taken up into biomass, decomposes from that biomass, and settles. What you're seeing over here is a picture of a lake experiment that was done up in Canada in the 70s and 80s. It's been years. And what they did, I don't think we can even get away with doing this, is this little pinch point, they erected a barrier. They literally put a weir all the way down to the bottom and dumped a bunch of phosphorus into this side of the lake and not into this side. And you can tell, even though the picture's terrible, that there is an enormous amount of algae growth up on this side and really pretty clear blue water at the bottom. So this is a whole lake experiment to try to look at these limitation behaviors. So from this experiment and many others in smaller and big scales, we've kind of understood that phosphorus is primarily the limiting nutrient in freshwater systems. So the phosphorus cycle is simple compared to what we see in, I don't know why that's on the top of there. Let me get rid of that. It's supposed to be down here. So compared to nitrogen, phosphorus is pretty simple. There's some that's attached to particles, but there's no gas form. There's a lot that's attached to minerals like soils and different kinds of rocks and things like that, and it's at various levels. So there's like primary minerals, surface absorption, other kinds of compounds. But for this course and for what we need to know is that there's kind of this like non-organic fraction or this mineral form. And that's how we actually mine it for access. This is what's happening in Florida. It's one of the largest phosphorus mines there, right? So we've got phosphorus. We have it as mineral form. It can be solubilized and we can have stuff that's available for plants. And then we have organic forms that are tied up in biomass of various kinds. They might be residues from plants. They might be solids from animals. But all of that is collectively called organic phosphorus. So we've got mineral stuff over here. We've got organic stuff over here. Both of those are pretty unavailable for plants to use. They need to be soluble for plants to use them. So they need to be turned into these solubilized phosphorus. And so it's basically like a phosphate ion. In this guy figure, it's kind of shown as a hydrolyzed version with the hydrogen on it. But it's fine. This is basically that soluble form. But that's what's allowed for plants to take up and to use in their growth. You could superimpose this on a water body like you're doing for your lab. And that is the same form of phosphorus that's used for biology. So this is a little more in depth of looking at a soil particle and how we think about its connections to availability. But phosphorus can be sorbed to iron and aluminum pretty strongly. And then it can also kind of live inside and be kind of part of kind of the structure, kind of like think of a sponge soaking it in. So it's kind of like a little harder to get. So you've got absorption on the outside, absorption like a sponge soaking it up, but it's still relatively loosely held. It's not connected to a compound. And then you've got these different more chemical reactions where you start to build more tighter storage because it's actually turning into a new chemical. So you can have a range. This is kind of a little more complex, but I think the takeaway is you can have a range of availability of phosphorus. So what we want for plants is this, but there's all these different ways in which phosphorus can exist on a particle that are either really available. This absorption, this stuff is like pretty quick to get rid of. But as you get further along, you get stuff that's really, really tightly held. And it's hard for plants to get access to it. And it's hard for it to be washed away. So this is just showing that there is a lot of different availability of phosphorus in soils.